When I began my career in medicine, I assumed cognitive decline was simply inevitable. With each passing year, I believed that mental sharpness, recall ability, and the speed at which the brain processes information would naturally deteriorate. As it turns out, that assumption was entirely mistaken. What modern neuroscience now reveals through the study of neuroplasticity paints a strikingly different picture. The brain is far from a fixed, unchanging structure. It is, in fact, a living and constantly adapting system that continuously reshapes itself in response to the inputs and stimuli it receives. The most efficient path to a sharper, more resilient brain is simply learning how to feed it the right signals. What follows is a practical, research-backed guide to the most effective methods for enhancing brain performance. We begin with low-effort habits that require minimal lifestyle disruption yet yield consistent, measurable gains. From there, we progress toward more demanding strategies that call for greater commitment but produce considerably more profound and enduring cognitive benefits. Starting simple. The case for a daily multivitamin. Perhaps the most accessible entry point is one that demands almost nothing from you in terms of time or effort, and yet the evidence behind it caught me genuinely off guard. A large-scale randomized controlled trial enrolled more than 3,500 participants, dividing them into two groups, one taking a daily multivitamin and the other receiving an identical-looking placebo, over a period of 3 years. Those who took the multivitamin consistently outperformed the placebo group on cognitive assessments, particularly tests measuring immediate memory recall. When researchers placed these results alongside established benchmarks for age-related mental decline, the multivitamin group appeared to have retained approximately 3 years' worth of memory function that would otherwise have been lost. Regarding the integrity of the research, the manufacturer of the supplement used in the trial provided financial support through a grant and supplied the pills, but played no role in study design, data analysis, or the interpretation of findings. The research itself was carried out by independent scientists at well-respected academic institutions. My conclusion from this evidence is that I now hold a much stronger position in favor of taking a high-quality daily multivitamin as part of a brain health strategy. This marks a significant departure from my earlier stance. I had long dismissed multivitamins as largely wasteful, believing most of the nutrients passed through the body without being meaningfully absorbed or utilized. This trial reshaped that view, particularly around memory preservation and the slowing of cognitive aging. The lesson here is that addressing subtle nutritional deficiencies carries far greater consequences than most practitioners had previously appreciated, and that this becomes increasingly true with age. Dr. Choline. An overlooked nutrient with rapid cognitive benefits. Another highly effective yet widely underappreciated tool for cognitive enhancement is increasing your intake of choline. This nutrient can produce noticeable improvements in concentration, memory retention, and mental processing speed relatively quickly, yet the majority of people fall short of adequate consumption. Choline is a vital nutrient that supports brain function through two distinct pathways. The first involves the synthesis of acetylcholine, a neurotransmitter that plays a central role in attention and the acquisition of new knowledge. Each time you attempt to concentrate or learn something new, your neurons rely on acetylcholine to relay signals between one another. Greater neuronal communication translates directly into stronger, more reliable memory. The second pathway involves the maintenance and regeneration of the cellular membrane surrounding each brain cell. This outer layer operates similarly to the protective coating around an electrical cable. When it remains intact, neural signals travel with speed and precision. When it degrades, signal transmission slows, and the brain's ability to form new memories is compromised. Fortunately, obtaining adequate choline through diet is not complicated. Eggs represent the most convenient and practical source, with 1 to 2 eggs per day meeting the needs of most individuals. For those who avoid eggs, alternatives include oily fish such as salmon, tuna, or cod, poultry and beef, and plant-based options like soybeans. While choline supplements are available, they are seldom necessary given how accessible dietary sources are. Clinical evidence reinforces this food-first approach. A randomized controlled trial conducted with Japanese participants demonstrated that consuming 300 milligrams of choline from egg yolk daily over 12 weeks, an amount equivalent to roughly 2 large eggs, produced significant improvements in verbal memory compared to placebo. Longer-term research adds further weight to these findings. A large prospective study carried out in Finland tracked approximately 2,500 men over more than 2 decades and revealed that individuals with the highest phosphatidylcholine consumption faced a 28% reduced risk of developing dementia relative to those with the lowest intake. Higher choline consumers also demonstrated superior performance on assessments measuring memory recall and verbal fluency, tests that evaluate the frontal and temporal regions of the brain governing information organization, verbal communication, memory encoding, and higher-order decision-making. Blueberries. Modest quantities, meaningful brain protection. A third dietary adjustment worth incorporating is the regular consumption of blueberries, a change that supports not only cognitive performance, but broader aspects of physical health as well. Among foods studied for their effects on brain health, blueberries stand out for the consistency of positive findings across diverse populations and age groups. Their primary mechanism of action relates to their high concentration of anthocyanins, the compounds responsible for the fruit's characteristic deep blue and purple coloration. These molecules have a particularly favorable effect on the brain. Anthocyanins stimulate the production of nitric oxide, a compound that causes blood vessels to expand and relax. As circulation improves, the brain receives a richer supply of both oxygen and essential nutrients, which enhances cognitive flexibility and responsiveness across a broad range of mental tasks. One of the most reassuring aspects of this finding is how modest the required quantity actually is. Approximately 1 cup of blueberries per day appears sufficient to realize most of the associated benefits, and fresh fruit is by no means required. Frozen blueberries and powdered blueberry concentrates have been shown to produce equivalent outcomes. In one rigorous, double-blind, randomized controlled trial, older participants who consumed 26 grams of freeze-dried wild blueberry powder daily for 12 weeks demonstrated measurable improvements in immediate memory recall and notably better performance on cognitive flexibility tests compared to the control group, with these gains emerging within just 3 months. Broader analyses of the research landscape confirm these results. A systematic review and meta-analysis, drawing on 9 separate randomized controlled trials and involving more than 500 total participants, found that regular blueberry intake produced significant memory improvements even in individuals already experiencing mild cognitive impairment, suggesting that blueberries offer value both as a preventive measure and as a supportive intervention once early cognitive changes have begun. Aerobic exercise, the most powerful tool for brain rejuvenation. We now turn to an approach that demands more discipline and physical effort than any of the dietary strategies above, yet surpasses all of them in terms of the magnitude and durability of its cognitive benefits. By most measures, regular aerobic exercise represents the single most effective anti-aging intervention available for the human brain. A highly influential study examining the relationship between physical activity and brain health, published in a leading scientific journal in 2011, randomly assigned older adults to one of 2 groups, one engaging in structured aerobic exercise and the other performing stretching and toning routines over a 12-month period. The aerobic group walked on a treadmill for 40 to 45 minutes per session, 3 sessions per week, maintaining a heart rate intensity equivalent to 60 to 75% of their individual maximum. The outcomes were striking. After 1 year of sustained aerobic training, neuroimaging revealed a 2% increase in hippocampal volume among the exercise group. The hippocampus is the brain region most closely associated with the formation of new memories and the ability to navigate physical space. Under typical aging conditions, this structure shrinks at a rate of approximately 1 to 2% annually. A 2% volumetric gain therefore effectively undid 1 to 2 years of normal age-related brain deterioration. Further analysis pinpointed the growth specifically within the anterior portion of the hippocampus, the frontal segment responsible for spatial memory functions such as recalling where objects were placed, retracing familiar routes, and orienting oneself within unfamiliar environments. The structural improvements were accompanied by equally notable biochemical changes. Exercise participants showed elevated concentrations of brain-derived neurotrophic factor, commonly referred to as BDNF, a protein that acts as a growth and repair agent for neurons. BDNF facilitates the formation of new synaptic connections, supports the recovery of damaged neural tissue, and underpins neuroplasticity, the brain's fundamental capacity to reorganize, adapt, and strengthen itself in response to experience. Rising BDNF levels are associated with accelerated learning, more durable memory formation, and a meaningfully reduced risk of age-related cognitive deterioration. The convergence of enhanced cerebral blood flow, reduced neuroinflammation, and increased BDNF production collectively explains why aerobic exercise exerts an influence on brain health that nutritional interventions alone cannot match. It acts directly on brain architecture and neurochemistry, providing the physiological conditions under which the brain can genuinely repair and fortify itself. While the study in question used treadmill walking as its exercise modality, any aerobic activity capable of sustaining a heart rate within the 60 to 75% intensity range will produce comparable results. Cycling, rowing, using an elliptical trainer, swimming, or jogging are all valid alternatives. What matters most is selecting a form of movement that you find genuinely enjoyable and sustainable, since consistency over time is the single most important variable in determining the magnitude of benefit you will ultimately experience.